Well, you made it. This is the final video in our Unit 8 series. We've covered a lot of different things in this unit, and you guys have been amazing in showing me your learning and your understanding, and I think the depth of your learning has really gone to new heights. So I think for all of you, you should really pat yourself on the back for a kind of a personal best in terms of academic achievement, I think, for almost all of you. So in any case, this little last piece here is just intended to kind of go through the different standards. We'll go through all 8 of them, whatever they were, to kind of give you that heads up on those little things that you need to remember to think about just kind of overall, and kind of what some of the things are going to be on the test and things you need to remember. So let's dig in. So very first thing we took a look at, other than mitosis. I know in mitosis we kind of did as a warm-up, and it's definitely wrapped into here, but I'm not going to test you super specifically on mitosis. We were just using that as a way to practice demonstrating our knowledge. So the first one we're really going to look at is heritable traits. That was objective 8.1. What's the difference between traits that are heritable or not heritable? What are acquired traits? And what are some of the interplays between the influences of genetics versus the environment? So you should be able to give some examples of each kind of trait, and you should be able to give examples of how, say, environment, either external environment or internal environment, could influence genetics. So by external, I mean something, literally external, like temperature or sunlight or something along those lines. Internal environment could be more like biochemical stuff on the inside. Like, for example, the way your hair color changes oftentimes as you age is genetics and environment, not just outside. So there you go. Second standard had to do with all of that vocabulary piece. So there's lots and lots and lots of words that we hit really early on in this unit, which probably by now you're pretty comfortable with. But if you're not, there's a couple that you really need to take a look at specifically. A lot of times students have trouble with the term allele, and figuring out what that means. I know you guys know dominant recessive, but how can you tell if something's dominant or recessive if I don't tell you? Make sure you know the difference between genotype and phenotype. Students a lot of times answer that question wrong, like, oh, I knew that one, because they were answering about phenotype when the question asked about genotype. And you guys are pretty good at homozygous, heterozygous from my scene. Application of the term segregation, independent assortment, definitely two of the hardest terms on there, but we've been revisiting those throughout some of the later standards, so hopefully that's a little bit clearer to you as well. Next thing that we moved on to was the Punnett square parts, right? We had a bunch of different things that we did with Punnett squares. You guys are fantastic at doing 1-trait Punnett squares. 2-trait Punnett squares, most of you are getting that one, or got it, I should say, at this point. But again, those are a little bit harder, so I would definitely go back and make sure that you are comfortable with sort of analyzing a story problem and then setting up a Punnett square that has 2 different traits at the same time. And then can you analyze genotype and phenotype ratios? And especially with dihybrid crosses, I'm only going to ask you for phenotype ratios, not genotype ratios. But can you tell me either percents or counts or something along those lines? There are some probably reverse Punnett square problems. Reverse ones, I mean, are the kind where I tell you, how is it possible for a child to have type O blood if mom's got type A and dad's got type B? Where you're starting kind of from some point that's inside the Punnett square and then kind of making assumptions about what's on the outside of the Punnett square. That's what I mean by reverse. I said, most of you are pretty good with that. Definitely double check your dihybrid Punnett squares and make sure you got those down. From there we moved on to kind of the funky ones, right? The different kinds of adaptations or the different kinds of modifications of heredity that we looked at. So incomplete dominance and codominance. Can you look at things like multiple alleles versus polygenic, sex-linked inheritance? And there was Punnett squares that went along with most of those. We didn't really do Punnett squares for polygenic. That's going to be more of an interpretation question. But all the other ones are definitely involved in Punnett squares that we've done. So can you also take a look at a story problem, and can you tell me which kind of inheritance it is? So like if I give you a problem that says, a brown cow breeds with a white cow and you get a beige cow, what kind of inheritance is that? Can you tell me that's incomplete or codominant or whatever? So make sure that you kind of look back through the Punnett square packet for some of those kind of little key bits of language that maybe would help you identify whether something's dominant or recessive or codominant, whatever. That's a little bit tougher. Going on from there, a couple of key things that you do need to be able to do. Make sure you know the ABO, the blood typing stuff, as well as the RH, which is positive negative. There will be, I think, a little thing on the test about, like that little blood typing worksheet, where if this one clumps up and this one doesn't clump up, is it type A or is it type B? Is it positive? Is it negative? There is probably going to be a dihybrid cross with blood types. We can do a dihybrid cross just like we do any other. Instead of big A, big B, little b, it might be, say, big A, little I, and plus minus. It's the same kind of process, but it can be kind of challenging to do that. The other ones that are a little bit tricky are doing like sex-linked ones, being able to deal with the X's and the Y's and what gets attached where. I may throw on one of those bird questions on there where they do the ZW, ZZ system. Same idea as sex-linked, so if you can do one, you should usually be able to do the other one. When you're talking about height and skin color and weight, kind of what would that look like? We generally don't do a Punnett square in those. We kind of get that rainbow effect. A lot of times there's like a little graph showing all the different phenotypes that the average phenotypes are. The graph kind of looks like this, right? Where you've got, not too much at the extremes, lightest, darkest, and then more as you get to the middle. So those kinds of ideas when you're talking about the advanced Punnett squares. And then we moved into meiosis. We started looking at the cellular basis for genetics and what were the steps of meiosis. So can you tell me what the names of the phases are? Can you use all that vocabulary, especially the new vocabulary like gamete, which is egg or sperm, haploid and diploid, and tetrad. Those are some key terms that you need to be able to kind of add into prophase, metaphase, anaphase, telophase. And you should be able to identify those key stages where differences happen, like in prophase 1 of meiosis, tetrads form, and in metaphase 1 of meiosis, the tetrads line up. But meiosis 2 is just like mitosis. Those sorts of things, you should be able to kind of identify those key similarities and differences. Then we move forward with that and took a look at what happened when things went wrong, maybe in meiosis, and that was the section on karyotyping chromosomal disorders. So you should be able to identify those major ones that we talked about in class. If I give you a picture like this one, can you identify whether it's a boy or a girl, whether they're normal or abnormal, and what the name of their disease might be? And then can you maybe even tell me that, well, hey, that has to do with a mistake that happened in metaphase to anaphase 1 of meiosis, that kind of stuff. And then we started into this kind of last sort of big theme, which is about the theme of variation. And so in 8.7, we were looking at the links between meiosis and our rules of genetics. Like, how is segregation demonstrated in meiosis? How is independent assortment demonstrated in meiosis? And then how can you get so many different kinds of gametes that form? And that idea of that 2 to the n rule, that you get as many different eggs or sperm gametes as you have numbers of chromosomes, or that's the power to the 2 to that power of number of chromosomes. So it's kind of crazy in terms of the numbers of possible offspring that you can get. And then the last bit was taking that variation question even further, and we talked a variety about some of the advantages and disadvantages of sexual and asexual reproduction in terms of energy expenditure, environmental matching, ease of finding a partner, guarantees of success, those sorts of things. And then you should be able to relate that idea of variation to success in a changing environment. That asexual is probably better because it's easier and lower energy when the environment doesn't change. But when the environment is changing or when it's unpredictable, that sexual variation and sexual reproduction and the variation that goes along with it is definitely an advantage over time. Not necessarily to one individual, but over time to an entire species, having that variation present in the species is definitely more important. And again, can you use those kind of important bits of vocabulary? Can you talk about independent assortment relating to meiosis and sexual reproduction? Can you talk about crossing over? That was kind of a big one in this one. The fact that chromosomes literally break off and exchange little pieces next to them, so the number of independent assortments is actually even bigger than we expected. Can you use the term mutation? Can you talk about non-disjunction and term fertilization? So there you go. There's our big topics that we talked about throughout the whole thing. We had a big transition through here. I think if you go back and take a look at your work that you've done, you'll find that because you took the time to learn it along the way, that reviewing for the test is actually going to be a lot easier than what you've done in the past. That means doesn't mean don't do it. You got to look back, make sure you're fresh, make sure you got all your vocabulary. But I want you to know that because we've checked in with your learning at every single step in the way, that you should walk into this test with some confidence. You can do this. And you've shown me that over and over and over again in your notebooks and things like that in class. So, all right, see you for the test. Tell me when you're ready.